I've been at Cal State Northridge since 2021, so this is my third year. And I worked with many different things in the past. And then when I created my lab at Cal State Northridge, I decided to study a few things that I'm passionate about: natural selection, human history, and ancient DNA, and tried to put all those three things together. The title of the talk is a little long, right? Like how ancient epidemics shaped the evolution of the human immune system. This is part of my research program that we already have some results to present. The whole research program is based on this idea where we currently have ancient DNA, and then we can assess, we can measure genetic diversity, how to say, analyze or assess genetic diversity in the past from the human side, but also from the pathogen side. And people have been working on those two organisms separately, in my point of view, as far as I know, at least most people. So some people have some ideas to understand how human evolves and what's the routes of peopling of the different continents and so on. And some people are doing similar things with the bacteria, the viruses, and other pathogens. So my idea was we have a lot of new data, a large amount of data for humans for sure. For pathogens, we have a good amount of data. And my idea was to see if I could actually time evolutionary gains in one species, humans, with the evolutionary gains of some event, some like emergence of a new strain of a bacteria or virus, or an evolutionary gain like a plasmid or some new gene that makes the bacteria evade the immune system, something like this. So if I could time those two things and see how those two things would be correlated with each other. And that was my way to define, the way I used to call that was host pathogen coevolution. I know people that study host pathogen coevolution, they don't like that I call this type of thing that way, but in my point of view, it is. And I hope you understand what I'm trying to say here, right? So there are two organisms evolving, and the responses, the evolutionary gains or the changes that we see in one organism will have an impact in the other organism. And I was curious to see if we could actually see that. The research is not finalized. I'm still a lot focused on the human and doing very little things with the bacteria. You will see towards the end. But that's the general idea. That's where I come from. And this is an outline of the talk. I have to say one thing. I hate when people like are giving talks and they have excuses. My computer, my bottle opened in my backpack yesterday. My computer was completely wet. I was afraid to turn it on for the whole night. And then I didn't have a lot of time to finalize the very few last slides. I like the first ones. They are nice, but the last ones, they have a lot of text. Sorry for that. But let's get there when we get there, right? So this is a general outline of the talk. I'm gonna start talking about natural selection and recent human evolution. I think most of you know the things that I'm going to be talking about here, but I'm going to be trying to already insert some concepts that I'm going to be using later, which is how allele frequencies change across time and how can we assess that? How can we measure that? Then I'm going to talk about how ancient DNA in humans have been traditionally used, which is mostly focused on understanding history, like different the peopling routes of different continents, for example. Then I'm going to talk about some attempts that many different people are doing to use ancient DNA to study natural selection. I'll mostly talk about like superficial theory, kind of like, I'm not going to go deep into the theory, but I'm going to be talking about mostly like what is the theory or what's the rationale behind those analyses. And I will not bring a lot of examples, but I will then talk about my example, my research, which is some of the results that we already have. We are already planning we are already working on a manuscript. So it is kind of finalized, although there are many other areas that we want to explore. So those are the four items. So let's start with natural selection and recent human evolution. When we study human evolution in general, we talk about evolutionary forces. And I know some people consider other forces also evolutionary forces, but I think everyone agrees that those four are evolutionary forces, right? So we are studying those four forces. Mutations are the ones, the source of all genetic variation that we see in any living being. Mutations can happen by errors in replication or endogenous damage, exogenous damage in the DNA. And that's the source of variation that we have. Gene flow, it's a way that once diversity appears in one population, it can be brought to a new population. So it's new for that population, but it's not completely new, right? It's just mutation generates that variation. Then there is genetic drift and natural selection. Both of them, once genetic diversity arises, both of them will shape genetic diversity in different ways, or sometimes in similar ways. Genetic drift will make changes, will result in changes in allele frequency that's going to be random. Sometimes you see some allele going in high frequency and fixating, sometimes low frequency and disappearing. But there is a sort of behavior that we observe, which is caused by this sampling error. So when you make a new generation, you are not representing every allele necessarily from the previous generation to the next. So because of that, we have this random variations. They are usually not very large, right? They are small. Whereas natural selection, depending on the strength of natural selection, we're going to see very fast changes. And even if when it's strong, when it's not strong, when it's weak, we also see some sort of directional change, as opposed to genetic drift that kind of goes all the way. I have some allele trajectories here from simulations that we ran with the starting allele frequency. I think it's 10%. And then we see in neutrality, you see allele frequencies in the end of the simulations, in the end of the 500 generations. Sometimes they are very low frequency, near 0 or 0, and sometimes they are a little bit higher, right? Higher than 10%, let's say 20% and so on. And as you start having weak selection, you see this type of behavior, but you start seeing some sort of direction. This is positive selection in the sense. So the frequency of these alleles has some sort of direction of increasing. And as you have more like stronger and stronger selection coefficients or strength of natural selection, you see this change being much faster. So we actually can learn about natural selection by observing this allele frequency changes, right? In humans, we couldn't do that before ancient DNA, because, but in other organisms that you could do experiments, like with bacteria and flies, you could actually observe those things directly. And then with the advent of ancient DNA, we were able to directly observe that. But while we still didn't have, and still nowadays, actually, people don't necessarily need ancient DNA to do this type of study. We use a lot of different methods. I'm just showing here two things. One is one that it's very used, which is FST based. Basically, it's gonna, FST measures shared drift, let's say, between two populations. So when you see those two populations in the top there, let's say half of the individuals are red and the other half is blue in one population and the other one is the same thing, you don't see a lot of difference between them. So you have an FST of 0. And as you go move down here, you have like 10 individuals that are red and 10 individuals are blue, like in the different populations. So you have FST equals to 1. So we can leverage that, like the genome will have an average FST when you compare two populations. And you can see if specific loci in the genome are outliers in this, based on this average or this expected distribution in the genome. And that by doing so, you find alleles that evolved in one population because of local adaptation. I mean, the allele frequencies changed very fast because of local adaptation, for example. The same thing here, once a beneficial mutation arises, it spreads in the population to the point that might be fixed in the population. And what happens here is that the you start to other mutations that are not beneficial necessarily, they might be neutral. They hitchhike with that advantageous mutation, and they increase in frequency as well. And then what you see is an extended haplotype homozygosity. So those are just two different ways that we can use present-day DNA at population level to infer natural selection. Okay, and those studies, this is published in 2016. I like this one. I was looking actually for a more updated one. If anyone knows, let me know. But this shows in 2016 a few examples of human local adaptation events, or let's say shows regions that we believe we have evidence that humans adapted to some sort of selective pressure. And there is the names of the genes here. It doesn't matter. What I think it's interesting is that you see all over the world, right? We see in South America, we see in Greenland, we see in Asia, Europe, Africa, etc. And the other interesting thing is that this type of event, natural selection, affects many different phenotypes or types of phenotypes, let's say. So lactase persistence, diet, height, Arctic environment in general, again, diet, high-fat diet, thick hair. And then there is something here about immunology, right? Trypanosome resistance and malaria, for example. More stuff related to diet, starchy food, and so on. So we start to see some patterns here when we look. If you have done these types of scans before, you start to see something that there's a lot of metabolic genes involving metabolism that are selected, but also a lot of genes related to natural selection. I think every single scan of natural selection that I ran, and the ones that I remember that other people ran, there is at least one peak in one immune gene. So that made me think, let me study that, let me try to use ancient DNA to try to study natural selection. What would be the easiest way to go? I thought immune traits. So that's one of the reasons I chose the immune traits. But I've been also studying other things like high altitude adaptation, for example. Anyway, so this is just a quick, like a broad picture showing that examples of local adaptations are spread all over the world. They involve many different phenotypes, mostly metabolics and immunology, but many others as well. And those are all indicated by single genes. I'm going to come back to this later. So most of these scans, they point to a single gene that is involved in adaptation. So for example, the lactase one is there, NCM6. That gene is close to LAC, I think, right? The gene for lactase, LCT. And so we know, oh, that's the gene that is involved in that phenotype. So many of those studies try to do that, and they do that successfully. Do you have any questions while I drink some water? Okay. So I talked a little bit about natural selection, some methods that we can use, and also some examples of studies that identified single genes related to local adaptation. Now I'm going to talk about ancient DNA, which is that other passion that I talked about, right? That I wanted to unite with natural selection. So ancient DNA in human history. Oops. This is a figure from 2021, Chao Mei Fu's paper. You can see the map here, right? So this is a map. I don't know if you can see here, like Brazil, South America, United States. You should be, right? You should be able to see. Can you see more or less like the map? Oh, that TV has the map. But this one doesn't, right? Yeah. Okay, so this shows a map, and it shows each circle means the number of, it shows the age of the different, no, wait. Where is the color code? it shows the size, the number of samples, but also shows the age of the samples that were collected. This is not very relevant. I just wanted to show like there is a lot of DNA. I counted yesterday 9,990 in one of the repositories that ancient DNA people are using. So almost 10,000, and not all every single sequence is there. So we have over 10,000 ancient DNA sequences. And the weird thing here is that they're all concentrated in one place. Where is it? Eurasia, mostly Europe, actually, right? So there is this geographic bias. This is a little bit of a distraction here. Doesn't belong to the talk overall, but I wanted to highlight that there is this type of geographic bias. And this is not only in ancient DNA. Actually, we have, I don't have the whole figure, sorry. So this shows, I didn't check it before, sorry. But there is a bar here showing the number of people that are living in each continent. And then you see that Europeans are actually, I don't remember how, but it's like a small piece here of this bar plot that was here, showing that the majority of people are not European, although the majority of people that are being studied by genetics are European. This is not ancient DNA, this is modern DNA, present day DNA. I just wanted to highlight that. There's a thing here. And yeah, so we see this bias, and this is horrible because you start knowing more about the populations that are better served, and then you can provide better service for those like precision medicine and so on, but just mostly for those that are mostly studied, right? So this is only makes the situation worse. But anyway, so this is what we see in present day, and there is no excuse for you to not study present day DNA from other populations. Ancient DNA, we are limited by the availability, right? So there might be an excuse, but still it's very biased. Anyway, despite the bias, we have 10,000 ancient DNA sequences or genome-wide data for 10,000 individuals from different time points. And this has shed light on many aspects of our history. For example, it showed that archaic humans like Neanderthals and Denisovans interacted with modern humans. We know the areas that they interacted, we know the time more or less, and we also know how that impacted our phenotypes. Also shows how the population's history or population dynamics of modern humans. I think, I might be wrong, but I think in Europe, we know there are three main groups that peopled Europe. And I think that information was not very clear from the archaeological point of view. I might be wrong in with that claim, but genetics can give us, confirm other point of views of other areas, but also bring new insights about history. There is also the possibility of doing kinship inference that you can actually do with a very high confidence using genetics, whereas with other elements you don't. So you can, for example, study the history of populations from the past and understand how they form families. Was it by biological link or not? Was something else? So those things are a lot of different things that ancient DNA is bringing for us. Recovery of lost genetic lineages. I like to put that here also, not just for those who study humans, but also people that study extinct species, for example. This is really interesting. And phenotypic reconstruction, people have used DNA to see like what's the color of the skin or the eye of some individuals. And then it evolved a little bit to things that are more complex, and I'm going to be talking about later. But ancient DNA has helped us a lot to understand our history and our evolution. And I think, at least as far as I know, let me see, I'm going to, just to see the time. Okay. As far as I know, I think this was the first paper to do some sort of natural selection, give some sort of natural selection insights using ancient DNA. I might be wrong, but this is 2015. At least it's the first one that I know of. And I thought it was very interesting when it came out. It came out with like 230 new sequences. It was a huge number. And they were also talking about some phenotypes. Here, this paper is showing in this filled colors, filled shapes here, we see a hunter-gatherer, which is the population or like the culture that was before agriculture in Europe. And then you see a lot of different agriculturalists that lived in Europe or migrated to Europe. And we have the steppe ancestry, which is a third wave of colonization or population of people that came to Europe. And we think that that's the wave that brought the Indo-European languages that we speak. And so this is showing what the frequency of different mutations. So you have the name of the mutation here, or the SNP, RS, and then a number, and then the name of the gene. I put those tags there. So those two there are diet and metabolism, lactase and FADs. And then you have pigmentation, 2 genes, and then immunity, this gene. So you see already that you can see some sort of allele frequency change across time. This is not necessarily related to selection, as we would learn later, because those migrations might have been bringing already different allele frequencies from the origins, right? So that could just represent a mixture and no selection. But it was a cool, interesting, in my point of view, thing that they did. And here, those dashed lines you see, it's present-day populations from Northwest Europe, Great Britain, Spain, and Tuscany. So you see that the frequencies are higher today, and they were lower before. So I think what they were trying to say here is that there is some indication that there could be some natural selection happening here. Later, this is a paper from 2020. There were, it's a very heavy theory paper, I believe is this one, that shows, this is the general idea of the paper, and that's the idea that I wanted to explore, which is you have a population evolving from up to down here, and then you have 4 time slices, right? Time 1, 2, 3, and 4. And here in the middle, you have an environmental change that it's represented by the change of color from white to color, and blue in the case. And then here you have this, the same thing here, right? Time 1, 2, and 3. We know that the change happened here. And then you trace the derived allele frequency of a specific mutation, and you see that it increases in time, and it happens around the time that there was an environmental change. So this is the basic idea of most people studying natural selection with ancient DNA. So can we actually do this? We actually can't. I found this meme, I don't know, like it says, are you two friends? And then the population genetics say no, and ancient DNA says yes. So I think it represents a little bit how I feel, at least, like the sometimes population geneticists, like more heavy theory, they have some issues with ancient DNA, whereas I think we that work with ancient DNA, we say, oh, we are doing ancient DNA, we are doing population genetics as well. But the thing is that currently, I don't know if it will ever change, but currently we have a lot of limitations with ancient DNA, and you probably are familiar with it, right? So ancient DNA, it's obtained from human remains, tooth, or bones. And there is a bunch of problems here, mostly related to the degradation of the molecule of DNA, fragmentation, degradation. There is damage that happens as well. There is a bunch of other things I'm going to skip, but there is a bunch of things related to degradation. And then there is also some bias in sample representation, which we talked about before, right? And I think those things hinder our ability to do some sort of something like this, because at most we have like one individual here, one here. So we know if the allele is there or not. We know if the allele is there, but we don't know necessarily if, well, anyway, I think I'm losing myself here, but we don't have a good sense of the allele frequency, right? Okay, so me and my colleagues, I was a postdoc at the University of Lausanne at the Malaspina's group. And I started to work with Lucas, who is a PhD student there. We started to work with selected some softwares. We selected 3 softwares. I'm going to tell the name later. And we started to simulate. He started to do that. That's mostly his work. I was mostly in the conceptualization of the work, but he's doing the work. So he's simulating allele trajectories based on either neutrality or different values of selection coefficient. So you will see, let me see, that first figure that I showed is his figure that shows a bunch of allele frequency changes. So he knows the allele frequency change, right? Because he's simulating it. And also he knows the strengths of selection that he's simulating. So based on those two things, what he did is that he got those three programs, ApproxWF, Lattice, and SR, which are from our colleagues. And they have different methods. And we started to use them with the simulations to see how they work. The problem with it, with this line here, so these methods were developed to handle time series data. Great, right? It's DNA, ancient DNA, and were tested with simulated and experimental data. I put microorganisms here. The ones that I know, that I remember, are with virus. So they had actually a good amount of data, and when they did the simulations also, everything was working fine. So however, their performance with ancient DNA remains a mystery. We were like, oh, can we actually use this or not? Because the data that they are using for time series is usually much better quality or like number than ours, the type of data that we use. This is a real data set that I'm working with. Those are ancient Peruvian. So basically, there is some years here. It goes from 0 present, and here's the past. So it's going to the present that way. And here's just like a random position. It doesn't matter the position. It's just showing that around 8,000 years, I have one sample. Around 8,500, I have one sample. Around 6,000, I have two samples, and so on. And then you see that it's very bad representation of the past. There's a lot of gaps. It's not very regular, the sampling, and that's one thing that we found out, that it's actually better for the methods that you have. I'm not going to show here, but to have like more regular sampling. And here, when you arrive in the present, you have a ton. We have many, right? We have 50 here, and then before European contact with Native Americans, we have like 500 years. Here we have a ton of samples as well. So those are recent. They are better preserved. Those samples are mostly from Peruvian highlands. There are some that are not from highlands. And that was the map there. So do these methods work with ancient DNA? They don't. We tried a bunch of different things, like different sampling schemes, different, the samplings are regularly spaced or not. The sample size in each point is the same or not. We tried many different things, and sometimes they work, sometimes they don't work. Here I'm showing, for example, that I think it's Lattice, the orange, is performing well for weak selection. And then when it's stronger selection, which is counterintuitive, it starts to not work anymore. So the real value are those red stars. And the program was run, I don't remember, it was like 1,000 times, and trying to estimate that. And then we have a distribution for each of these runs for each program. So we see that all those perform very well in this like quick selection, but as things get worse there, stronger the selection, we start to see completely weird behavior. Lattice has a straight line, and the other ones go estimate as near zero, neutral, whereas selection is much stronger. So here's just to show one of Lucas's graphs showing that those methods don't work for the type of data we have with ancient DNA, which is unfortunate because that hinders our application use of ancient DNA to study natural selection. Before moving on, I just want to say one more thing that ties to my research, which is this bias in sample representation. One thing that we saw is that ancient DNA and modern DNA, we have overrepresentation of Europeans, right? So this is another figure, but this shows the same thing, and now it's showing the map. You see all those red dots, they are European, and then we see very few outside. This is 2016, changed a little bit, but still very concentrated there. But when we look in Europeans specifically, we see that there is an underrepresentation of the blue here, which is less than 2,500 years before present. So it's more or less the historical era, right? Like Iron Age, I think, and antiquity, medieval time, and so on, the Roman Empire. So there is a lack of those samples, or like individuals coming from that time in Europe. And that was one thing that I wanted to explore in my research. And I started to explore that in 2017, I studied the barbarian migrations that were one of the topics that interested me in the Middle Ages. But not only that, the Middle Ages, it's very surprising that it took so long for people to start studying the Middle Ages, because we have, at least in the pathogens, they were already studying. But in the human side, no one really cared. And it's very interesting because first, interesting, I mean, it's sad, right, that they had this pandemic in the past, but we have several different outbreaks of diseases that happen in the Middle Ages that we could be studying, right? And we don't have much data for, at least from the human side until recently. We have also the invasions of the Roman Empire by the barbarians, or like interactions between barbarians and Romans, which is a topic that I also interested in, I studied. And this book here is called The Myth of Nations. It's by Patrick Geary. It talks about the origin of the different nations in Europe. It actually says that the creation of a nation in Europe is kind of arbitrary. There is like a lot of different reasons people created nations, and it's something that actually doesn't exist in fact. So anyway, super interesting, but a lot of the European countries that we know today, they have the origin in the Middle Ages. So come on, it's really cool, really interesting period, and it's understudied. Here is just another showing here the many diseases that happened in this medieval period. The most common known one is the Black Death. People know also the Antonine Plague and the Plague of Justinian. So just showing here that really cool, really interesting period to study. From any perspective. Any questions? Well, okay, so we want to study that. Talked about natural selection. I talked about ancient DNA to reconstruct history. I talked about that I would like to use ancient DNA to assess this time trajectory, allele frequency trajectories, right? To potentially be able to use those programs that tell you the selection coefficient in one gene. So we are doing an effort, it's a team effort here. Flavio De Angelis is a postdoc in my lab, and then Lars is my collaborator. I don't have an ancient DNA lab, so we do our analysis there. Betsy was 3 months with me, a postdoc between 2 jobs. So she did some of the analysis, and she still collaborates with me, mostly focusing on the pathogen side. And then Leah is, she's a bioanthropologist, and she has access to some collections in Europe that we are studying. So this is currently what we have. We are generating most of, like half of those individuals. We have about 350. We are generating data for all of them. We have mostly in Europe, in Italy. We have from the different periods there, you can see since Mesolithic, Neolithic until medieval times. So we are planning to have some sort of good time series for, at least for Italy. And then we have also one side in Britain from the Middle Ages, and in Romania we have from, it should be, Roman period, is correct, yeah, from the Roman period. And if you're going to the AABAs, I'm going to be talking about, poster or talk about this site. Anyway, so this is what we are doing. It's funded by NIGMS grant, and that's what my lab been doing for the past 1.5 years. So how we use this data, or how we plan to use this data to study host pathogen coevolution or pathogen-driven natural selection. I think I talked a little, I talked about this before, right? Ideally, in the end, I want to do something like this to see how humans evolved and see how the pathogens evolved, specific pathogens or like several pathogens, to see if we can see some sort of correlation between evolutionary gains in the 2 species, or different types of events in the different species. And this actually has been done similar to the idea that I initially had with, it was published a year ago, Klunk et al., 2022. Basically, they have here the population size in Europe. So it increases, and then there is the Black Death there, and the population decreases, and then it increases again. So there is a moment here that you have this this infection, this outbreak of Yersinia pestis, and it kills a lot of people. And the idea is that some people had was to compare allele frequencies before and after the Black Death, which is before Black Death, after Black Death, and modern. So this comparing those different moments in history to see if specific alleles were changing in frequency very quickly. Very cool. That's what I've been talking until now for 40 minutes. I have 10 minutes to finish. So that's what we've been thinking about. And this group came and did this, and then they found a bunch of genes. I don't know if you follow the discussion, but there was another group that wrote that found 4 methodological issues with this paper. They didn't replicate a lot of, there's a bunch of methodological issues, like statistical issues. I forgot which one. I know there are 4. So anyway, there are issues, but the idea is very interesting, right? And but they did that already. And then I was super upset because that was what I was planning to do, specifically with the Black Death. I was very frustrated. And then my postdoc is, he's a bioanthropologist, but he also did one of his postdocs with a medical psychiatric person. So he knows about GWAS. So from now on, I'm going to be talking about polygenic traits. And that's not something that I'm very familiar with. I'm still learning. So I'll try to not say anything wrong, of course, but I might not be able to answer many of the questions. But anyway, so here I'm comparing what's a monogenic and a polygenic trait. Basically, I'm going to go a little fast because the time is running out. So monogenic trait is a trait that is determined by one gene. And then we have polygenic trait, which is determined by contribution of several mutations. Usually we have a large mutation, a large effect mutation for the monogenic, and several small effect mutations for the polygenic traits. So my postdoc was saying, hey, let's look at the polygenic traits. It's much better, actually. And that's what actually he wanted to do. And here's this cool paper, 2005, I think, from Ryan Hernandez and colleagues. Classic selective sweeps were rare in recent human evolution, which is this type of, includes this type of monogenic selection. So there is this paper already, kind of old, saying that those are rare. And they are, and we have evidence that a lot of the adaptation that happen in humans, or a lot of the phenotypic evolution that happens in humans and other species, involves several traits. Oh, sorry, several genes. So, okay, let's do this that we were planning to do before, but using polygenic traits. So what should we do? Should we just check the changes in all the allele free? What should we do? Actually, Flavio's approach, he came with this idea of, let me skip this, to use what we call GWAS. So let me actually talk about this. So there are monogenic diseases, right? Cystic fibrosis, sickle cell anemia, Tay-Sachs. You might have heard of at least one of those. So those are caused by one gene, mutation in one gene that disrupts the function of one gene. And those are recessive autosomal, so you need both copies of the gene to be affected by the mutation. Whereas most of our traits are actually complex traits. They involve several different genes, and they also have a lot of influence from the environment. And they are usually characterized by hundreds, sometimes thousands of genes that contribute, each one contributes a little bit to the trait. Like the one that we most know about is height. I forgot the number of genes, but it's like a few thousand genes. Forgot 10 or what, but there's a lot of genes that are influencing height, but each one very tiny contribution. There are, of course, some monogenic diseases or monogenic traits that affect height, right? But typically height is determined by several genes. And so the way we study that is by doing GWAS, or genome-wide association studies. This is the definition here. I can share this with you if you're interested. Just going to go over quickly here. But usually, one type of design of GWAS is you have cases and controls. So when you compare cases and controls, the cases here are with like the hands up, the controls are with the hands down, just to show that they have different phenotype. And then you see some variants there being carried by the unaffected individual. Here, only one variant, let's say, could be either blue or red. You here see, I don't know, 20 individuals, 30 individuals, only 4 have. Here you see the same number of individuals, but many more have, like 3 times or 2 times as much. So based on like a statistical test, you can point that this gene is that carries this variant is associated with the trait. So this is oversimplification here, right? Because of time limitations, but basically you see some genetic variants between cases and controls. You can also do with things that are like continuous variables, like height, but let's think about cases and controls like this way. So what's important here to say is that GWAS inform us about correlation, not necessarily causation, okay? So yeah, that's very important. And I have another meme. What's the other meme? Anyway, I think I bring the meme later. So we have the GWAS that identify several loci, and they also identify the contribution of that loci to the trait. So I could have, for example, a mutation. This allele in this gene is going to give me 1 centimeter height. So if I have 2 copies of that, I'm going to be 2 centimeters higher than the person that doesn't have. So we always think about these effects as being like relative to other people that are in the population, right? So based on those small contributions that each mutation has to the trait, you can add all those effects and make a polygenic risk score. And here's just an example. So if we calculate here the trait, whatever, each person here has different variants, right? So if we count if we measure the polygenic risk score for each person, we can make a distribution out of that. And then we can find individuals that have low risk for the disease, let's say, for example, that carry only 2 variants, and someone that has higher risk that carries 7 variants, for example. So for the interest of time, I'm going to move. So we have GWAS and we have the polygenic risk scores that are a number that tells you what's your risk to develop a certain trait. We say risk, but could be like height or something that doesn't involve risk, but just like what's the probability that you have that phenotype. What do we do with that? So someone already studied the monogenic selections, right? So what we did is that we went to European data biobanks, like the UK Biobank and the FinGen, and we downloaded their some area statistics, and we found a bunch of different traits there that we were able to use that information to calculate polygenic risk scores. So we got that model, the polygenic risk score model, and we calculated for a bunch of Europeans from different time points. So for each time point that we use, like we have 200 and 400 time slices, we see some sort of distribution. I'm going to show the distributions in a second. Here I'm talking about some of the issues that we have. I'm going to go very quickly here. We select traits that are heritable, highly heritable. We end up by having those 7 traits, some coming from UK Biobank, some coming from FinGen. And we also checked for the correlation between the traits because some of these traits actually share a lot of variance with the others, right? So we want to, when we do the analysis, we want to know that we're looking at one trait only and not, and 2 different traits and not the same trait, slightly different. So, so we did all those things to sort of quality control. So this is our, what we did. This is some details about our data set. Basically, what I'm going to say here is that we got this data, this public data from Western Eurasia. We focus on 1.1 million autosomal variants. And the sample size that we have for different analysis is a different number, but the minimum number that we have is 2,400. And then we get those 2,400, and we group the individuals in time periods of 200 and 400 years intervals. Okay, and this is what we see. Ooh, it doesn't look so there. You see the concentration of the dots, right? So, and you see the mean, the median also. So you can imagine there is a violin here, or you can see there. But what we see here is that this is 400-year time intervals, and what we see here is that the mean slightly changes, right? Around this time, it starts to increase the mean of the polygenic risk score for one of the traits. And also it's over dispersed relative to the other ones. So there is a few differences here. And for the interest of time, I'm going to also move a little faster. But basically, we have 4 traits after a bunch of filters that we do and quality controls that we focus on that are actually giving us good information. And we do this time series analysis. The television looks better, so a bunch of these violins, and then you can we can do a bunch of different statistical tests to see if those distributions are different. And what we see is that these 3 time periods, when we do 400, but also when we do 200, which is like smaller and more specific, we see that these 3 periods here are the 3 periods that actually we see the most changes in the distribution of polygenic risk scores. So it's telling us that something happened there that affected these polygenic risk scores that were calculated based on immunological traits. So what we are seeing here is actually what we believe we're seeing because it coincides temporally with those 3 major outbreaks in Europe. You will see that those are potentially viruses, right? And our traits were focused on bacteria, but it potentially is telling us that there is a common response of a common set of genes that responds to infections. And we see this disturbance of the distribution of the polygenic risk score. We see the mean goes higher and the distribution is over dispersed. And this time, it's timed with the arise of those 3 major outbreaks. So yeah, so let me skip. Basically, that's what I wanted to say. So what we are doing here is starting to do, we're not the only ones doing that, but we are starting to try to get this knowledge from human genetics of polygenic traits, GWAS, and bring that to anthropology or human evolution using ancient DNA. And I think we were successful in the sense that we see statistically significant differences between time periods that have an outbreak, a known outbreak, and ones that don't have. We don't see that associated with the Black Death, though, which is surprising. But we see with the other ones, and I will stop here. And I want to thank Stone and Jennifer Vasquez for invitation and also for assistance with the visit. Everyone here for the audience, of course, my collaborators, in particular Flavio De Angelis, who did all this work. A little bit was Lucas, right? But he's doing all this work. He's creating this framework. It's very exciting. And also funding that's allowing us to do this research. And with that, I will have 5 minutes for questions. Thank you.
Questions?
Thank you for a really interesting talk. You may have said, and I apologize if I missed, what was the trait for the polygenic risk score that you were looking at?
We started with like almost 100 traits, but we went filtering for a bunch of different things, and we end up with 7 traits. Those are the 7 traits. I forgot why we dropped 3 of them, but in the end we have 4. Those 2 are included, and I forgot. Oh, so this is other bacterial agents classified to other chapters. Bacterial infections, and then I remember diarrhea and gastrointestinal infections is another one, and I forgot what's the 4th, but it's one of the other ones there. So they are all like related to. We got anyone that would have anything related to pathogen. Could be like predisposition or frequency that you had too many, you had little, anything related to that. And we went filtering based on heritability, based on if they were correlated or not, and etc. And we end up with these ones. They might not be the perfect ones, but those are the ones that have information.
Did you combine all the genes from these 6 or 7, or did you look at or the 5, I guess, that you're showing here?
So this is FinGen. So we have those 5 traits, for example, and we know for sure, for example, that diarrhea and gastrointestinal infections and intestinal infections are correlated. So what we do is that when we analyze one, we use the polygenic risk score of the other one as a covariate in the model. So if we still see differences, even controlling for that, we know that that's the specific trait that we are looking, that it's related to the adaptation or response to that pathogen.
So was it one of those in specific that you found that was associated with the infections, or
yeah, so
combination of them?
This one was, for example, this I know for sure. And the other 2 from UK Biobank we also found.
Thank you for the talk. Going back to your model, I think the model that Lucas created possibly, you were showing that weak selection coefficients. Oh, actually, I think it was a little bit later, but with weak selection coefficients, it predicted more accurately than stronger. So using this type of work, could you still use it when it predicts that the selection coefficient was weak, and you can assume that it's then more accurate, or just since it's not accurate for all possible selection coefficients, you wouldn't use it?
So the interesting thing here is that we did also different time intervals that we grouped our samples. So this one is actually a very large time interval. So we include, I think it's 500 generations or something like this. And because of that weak selection, we have a long time, right? So those small changes you detect better. Then when you have a very strong selection, the allele frequencies change very fast, and you group everything to get in one bin. So you lose that signal that you would see if you break that bin in small bins. So basically what happens here is that depending on the way you sample, you see better, stronger, better, weaker. So I honestly don't know how I would interpret anything, like unless I know the simulations, I know what's happening, but other than that, it's anything that you do, you see like something odd like this. This is counterintuitive, right? But as I mentioned, like because it's a large time bin, those small changes are detectable, whereas the other ones are not. So it does make sense after you think about. So it's all like this, like there's always an issue. Yeah. It's not that they are bad, right? But I mean, they can help you. I think they can help you maybe, but yeah, it seems a little bit difficult at this time to do like monogenic, study monogenic selection.
Thank you for the talk. I have a kind of similar question. I was wondering if this selection, if this simulation were under an equilibrium population, or did you consider like the demography of the population as well to compare with the selection?
So I know sometimes in simulations we do, we'd let the simulations run until the population gets in equilibrium. We don't do that here. Actually, I never thought about if it's wrong or not to do that, but we don't do that here. We just start with the 10, but the population has a 10%. I mean, the population has a constant size. Yeah, in this case, it has a constant size. I don't think we tested ever with size change. Our focus in it's South America, Peru, and also like the demographic history is not very well known. I think like we were, you know what, let's just do one population size and let's see how it works. If it works bad, I think it's going to work worse when you have a demographic history. So we just didn't even go there.
questions? Basically. All right. Well, let's thank Eduardo again.
Okay. Thank you so much. I didn't put my contact info, but if you, because of that issue with the water, I was like in a rush, I forgot to put, but you can find me online, I guess, and you can send me a message if you have any questions or comments.